well, thanks ever so much for turning out on such a glorious evening to come and listen to a talk. It's amazing that you have done so, but thanks ever so much. My name is David Thomas, and I'm the director of the Circumary National Research Network for Low Carbon Energy and the Environment, which I guess is hosting tonight's event together with the Sustainable Places Research Institute here in Cardiff. And I just want to spend a few seconds introducing what the NRN-LCEE actually is. We're funded through Welsh Government and HEFCU to the tune of about £7 million, and we've been given this £7 million to stimulate new research in Wales in the very broad theme of low carbon energy in the environment. We have a number of partner organisations who all signed up or who all put the proposal together to Welsh Government, but we do include all higher education institutes in Wales and all research institutes in Wales that have some interest in low carbon energy and environment research. Primarily, how we're funding our research is actually we've set up 8 consortia, or clusters as we call them, who are looking at research from marine renewables, so tide and wave energy, through to cleaner cows and sustainable soil management as well. So we do cover the whole sphere of that kind of research. And if you're interested in finding more, there is some stuff on the website, of course, but we do have some small booklets here at the front which actually describes the research that is going on in our clusters. There's a very notable one that is actually stemming out of this building, which is called Plants and Architecture. The Cardiff PI is Phil Jones, and together working with colleagues in Aberystwyth. So there's a couple more that Cardiff is involved with as well. So I don't need to say very much more about who we are or why we are here. It's clear that the speaker tonight, Sue Hartley, is right at the heart of the kind of research that we're interested in funding and bringing forward in Wales. In a minute, Terry will introduce Sue to you. The only thing that I do have to say is, as a practical one, if you could just sign in for us, that would be really helpful. Just pass this back, and I think that's it from me. So I'll hand over to Terry, who's over here. I'll hand over.
Thank you.
The microphone as well.
Oh, right.
Yeah, just.
Okay. Well, hold on. Thank you very much. It's a great pleasure to welcome you here and host this event at Cardiff and the Sustainable Places Research Institute. And it's a great pleasure to introduce Sue Hartley to us. I'm very struck by the fact that in Wales, with the NRN network and other infrastructures, one of the things that we're trying to do clearly is to link, and I think this is particularly critical, energy and food and the environment. The task here in many respects is to link these things together. In the end of the day, it is all about energy, but it's also about food. And this programme is particularly important in that regard. Sue has got an international reputation in the area of sustainable and resilient food production and protection, and her work is also looking particularly, I think she's going to talk about this evening, around the question of pests and diseases, which demonstrates that there's we're losing and potentially losing more than this actually with climate change. Something like 30% or more of food that's actually in the ground that we produce gets lost. And where I come from a social scientist, I also know that we're losing 30% or more from waste. So if you think about this, the efficiency or thinking about new forms of efficiency of food systems is a critical dimension which we've got to get right or get better, and applying interdisciplinary social sciences and sciences more generally to this is really very important. And Sue, for the last few years, interestingly, in parallel lives, Sue's been running been director of the Sustainability Institute at York, and meanwhile at Cardiff we've been developing that since 2010 in a similar sort of way. So the quest for bringing together interdisciplinary approaches to these questions, but also the challenges of linking energy and food together is particularly challenging. So we really look forward to hearing you, Sue, and welcome. Thank you.
Well, thank you for coming along this evening, and I want to talk to you about the challenges we face in feeding a growing population in a warming world. And a particular challenge is us having to do that in a sustainable way, so without causing environmental damage, or at least any more environmental damage to our planet. But I'm also going to talk about solutions. So talks about global challenges can be a little dispiriting if we don't think about ways that we can actually address some of those challenges. So it's not all bad news. But I think we are going to have a change in approach. And one of the things that we've already heard about in our introduction is the importance of interdisciplinarity, of people from different disciplines coming together. And that's very much what we're about at YESI, the York Environmental Sustainability Institute. And there we're trying to bring together physical, natural and social scientists, because it's that kind of collaborative thinking that's going to be needed if we're going to solve some of the challenges that we face. So we need collaboration between disciplines. We also need collaboration between countries. Starting to sound quite challenging already, isn't it? Now, global challenges need global collaborations. And I think, as we've also heard in our introduction, the importance of connecting the dots between these issues, because they're all connected: climate, poverty, energy, food and water. They're all interlinked. So we have to try and tackle them together, and we have to tackle them globally. And we have to get started because time isn't really on our side. We're facing a kind of perfect storm with some big demands on our slightly crumbling planet there by 2030. So we're going to have more people, we're going to need more water, more energy, more food, and we're also likely to be facing increased infectious disease burden. And we're doing all that at a time where the climate, if we're lucky, is going to be 1 degree warmer and may in fact be more than that. So one of the first things that presents us with is the need to feed more people from the same amount of land. As Mark Twain used to say, invest in land, they ain't making any more of it. And sadly that's true when it comes to agricultural land, and we have to feed more people than we do from the same amount of land. So at the moment we're feeding about 4.5 people per hectare of cultivated land. That's going to have to go up to, well, certainly more than 5 and perhaps nearer 6. I keep forgetting I can move it with this. So there's a bit of a gap between what we currently produce and what we're likely to need to produce by 2030. And the reason there's a gap is for 2 reasons. One is there's more of us, but actually that only accounts for about 1/3 of the increase in calories we need to generate. So we need to generate more like 15,000 trillion kilocalories a year, and we're only at about 9.5. And 2/3 of that increase is because of changing diets. More people want to eat beef burgers, or even horse burgers, or whatever is in a burger these days. And as people get wealthier, they eat more and they eat more meat. And that is harder a less efficient way of producing food because you have to grow the grain to feed to the cattle, then you eat the burger instead of just eating the grain. So about 2/3 of our increase in calorie need comes from our changing diets. And we can't put any more land into cultivation, not very much. We can't really increase cropping intensity in most of the places for reasons that you'll see shortly. So it's going to come from yield increases. So you're probably thinking, well, there's no need to panic because actually, haven't we kind of done this? Haven't we produced a lot more food? And it's certainly true. We're feeding masses more people than we used to. You can see this is the change in agricultural production from 1960 to now, and this is the change in cropland. So we've had hardly any more land in production and a lot more food. But we've done that through the so-called green revolution, through more fertilisers, through better varieties of crops. But the reason that people are worried, amongst other reasons, is that that pace of change has slowed dramatically. So the increase in wheat yields back in the day used to be about 10% a year, but now flat, down to about 0.8%. And wheat yields have flattened. We're no longer producing more wheat. It's just gone plateaued, basically. So we're not producing the same level of improvements as we used to. And yet we've got big drivers for change. So we need to address climate change. And in contrast to what our former Secretary of the Environment used to say about climate change, how good it would be to grow olives in Kent, in actual fact, climate change is not good for agriculture. So this is the FAO's, the UN's Food and Agriculture Organisation's prediction for changes in crop yields by 2050 if there's a 3-degree rise in temperature by then. It may be slightly less than that, but and you can see red means less crops, not more. Australia, most of Africa, most of Latin America, much of North America will see lower yields. Only some of the people causing the problem, northern US and northern Europe, will actually see increase in yields. And we can start to pick up those impacts of climate change already. So this is a study that came out in Science in 2011. An American team looked at the change in maize and wheat yields in different areas of the globe linked to the changes in climate and compared what the yield would have been if those changes in climate hadn't occurred. And they can already start to detect some rather worrying trends. So this prediction is already becoming obvious. So why is higher temperature a bit of an issue? Largely because of water. So when plants are growing at higher temperatures, they tend to need more water. And at the moment, global water use, about 70% of all the fresh water we abstract in the world, we use for growing crops. So there's not a lot of spare capacity there. And this is another cheery prediction from the Food and Agriculture Organisation. It must be a joy to work at FAO. And this is predicting where there will be water stress in 2050 and water scarcity. And unsurprisingly, Australia features, sub-Saharan Africa. And just keep an eye on northeast India, actually, because we will be returning there, well, metaphorically, not literally, later in the talk. And I've talked about interdisciplinarity and the importance of working with different disciplines, and there's very much a social science and political and economic element to this, because shortage of food leads to rises in prices. And price rises in many parts of the world are associated with political instability. So this is yet another FAO statistic or graph where they've charted the change in food prices over a number of years, and you can see where there are sudden spikes in food prices, there's a lot of political instability. All these red lines are riots or coups or political violence. So it's not good news generally in the agricultural world at the moment. And the former government chief scientist reckons we need to double food production but use less water than today. And we have to start now because millions of people are going to suffer if we don't. So I said this wasn't going to be depressing. You're probably thinking, if this is an upbeat talk, I want to be somewhere else. But just before we get to the upbeat bits, we'll just have a little bit more doom and gloom. And I wanted to introduce some of the key issues that currently agriculture faces. And one of our challenges is that we have a dangerous reliance on agrochemical inputs. So here are three big players in the food production world: rice, wheat and potato. And if we don't tackle pests and diseases and weeds, this is the loss of the potential yield that we get. So about 3/4, well, at least a half, but maybe 3/4 of the crop will be lost compared to what we could grow if we don't deal with pests and diseases. Now, if we throw our best efforts at it with mechanical, biological and chemical control measures, whether that's somewhat dangerously sloshing it on, trying not to breathe it in, or spraying it all on from a plane, we still can suffer up to 40%. So crop protection is helping. It's preventing the loss of quite a big chunk of our food production, but we need to do better, and in any case that isn't sustainable, because those chemicals, the pests are developing resistance to them, and we're losing the ability to sell them. A lot of them are extremely toxic, so they've been banned. We just need to think about the neonicotinoid pesticides and bees, so that's another agrochemical group that's off the market at the moment. And we've lost about 3/4 of our current pest control chemicals. They're no longer effective, we're not allowed to sell them anymore, but we aren't producing new ones. And if we could change these figures by about 1%, if we could save 1% of our crops from the mouths of insects, then we could feed 25 million more people a year. So it's worth trying to address that particular challenge. So why are our crops so pathetic? Why are they so vulnerable? Why do they need so many chemicals to help them? Well, the reason is they've been disarmed by domestication, is one way of thinking of it. So as we've changed our crops over the centuries, as we've bred them for being bigger and better and juicier and tastier, we've actually removed the traits that used to protect them against pests. So this diagram here shows the changes that they've been over the years, from 10,000 years ago when we first started to change these crops a lot, and the rate of change in particular traits. And we've changed lots of things about them. We've changed their life cycle and their reproductive strategies and their seeds and so on, but we've also changed this black line here the most. And that line is the changes we've made in so-called secondary metabolites. And those are chemicals that protect plants against pests and diseases. They're toxic, they're bitter, they're often brightly coloured. We don't like them very much, so we tend to remove them from our food. But unfortunately, that means that food is more vulnerable to pests as well. So anybody got any idea what these are? No guesses?
Boiled ones.
Sorry.
Boiled ones.
No. So I think
Potatoes.
potatoes. They are indeed potatoes. Proper potatoes, wild potatoes, potatoes that haven't been tinkered with, and these are Sainsbury's potatoes. So we like these a lot better, but of course so do all our insect pests and diseases. So we've made our crops a lot more vulnerable, and the other thing we've done is we've got rid of most of our crops. We only actually grow about 12 plants these days. And in fact, it's fair to say we probably only really grow 3. Wheat, maize and rice are 50% of the world's food production. So, and of those, as you'll see in a moment, we only tend to grow 1 or 2 varieties of those. So we've really honed in on 1 or 2 particular plants. And that means that there's very little vulnerability, very little resilience there to any kind of stress, because we're just reliant on 1 or 2 varieties of 1 or 2 plants. So cultivated Asian rice is the food of about half the world's population. Relies on Asian rice for most of their calories. So it's absolutely vital plant. But we only grow 2 kinds of rice out of 23 wild species. And we really, I was just trying to use the point there. We really only grow one variety of that really. So Rhizosis japonica is the most common cultivated variety, and that has 10,000 fewer genes than its wild relative. So we have absolutely stripped out loads and loads of beneficial traits. More on what we can do about that in a moment. We will get to solutions to some of these shortly. And in fact, we're going to start thinking about solutions right now, because we might have tinkered with our crops an awful lot. So this is maize, wild relative of maize. So it doesn't look terribly juicy, looks a lot nicer there. And of course, this is the evolution of wheat, and it's an acquisition of 3 sets of chromosomes. So we've done an awful lot of tinkering over the years, but fundamentally, our crops are grasses. The same plant family as on your lawn and the prairies, and that plant family is a well-defended plant family, actually. It has a secret weapon, not all that secret, because I keep telling everybody who'll listen about it. Silicon. And silicon is a very abundant element in soil, the second most abundant element in soil, and most plants don't contain very much silicon. So brassicas, like oilseed rape, don't contain very much at all. Daisies have a little bit more, but grasses take up an awful lot. And there can be some of the best plants that accumulate in silicon, like rice. Rice is a so-called hyperaccumulator because it can take up to 10% dry weight of silicon. So 10% of the weight of the plant is this element, far more than the protein in it or anything else. So what is rice doing with all this silicon? Well, it's a protectant for plants against a lot of different pests and diseases and stresses. So it's been shown to be beneficial for protection against herbivores and pathogens, and also drought. So I'm going to show you a little bit of work on that in a moment. So it's taken up from the soil in its soluble form, but it isn't by its nature very soluble, and it's deposited as these solid little granules, and they can take a number of forms, and you'll see lots of pictures of those as we go through the talk. So here's one that looks quite kind of round and smooth. Here's one that looks like a kind of mini razor blade, and here's another one that doesn't look exactly tempting if you're a herbivore. And these spines here are on the surface of a grass, and they don't look terribly appetizing either. So we don't like the look of those. What impact do they have on pests? So I'm going to show you some of the early work that I did on silicon and its effect on herbivores. And we started work on 2 very voracious crop pests. This is
It's fine.
Right. I don't recognise that slide. So this is African armyworm that feeds on cereals, and this is desert locust. And we took 5 different grass species and we grew them in low silicon environment and in a high silicon environment. So we could produce plants with low silica content, which are the black bars, and high silica, which are the white bars. And then we looked at which kind of plant these pests like to eat. And you can see that they ate a lot more, this is the amount they ate, and they ate a lot more of the low silicon plants. And they tend to avoid the high silicon plants. So it seems there's something about silicon that the herbivores don't like very much, and they moved on to feed onto the low silicon plants. So this was a deterrent effect. But when we looked at another pest, this is a grain aphid. This is quite a pest these are tropical pests, but this is a pest in Europe. And we found that the aphids weren't terribly bothered, actually. In fact, they seem to quite like, for one of the species, the high silicon plants. And we realised about 10 years later, because we weren't terribly quick on the uptake, that that's to do with how silicon is deposited in the surface of the leaf, which I'll show you about in a moment. So then we tried some slightly cuter herbivores. These are voles. And this time, just to keep you on your toes, the high silica plants are the black bars, low silica the white ones. And you can see that it wasn't quite as clear-cut as with the insects, but generally the voles prefer to eat the low silicon plants. And then we tried it with some very clever herbivores, sheep, And actually they're not quite as dim as they look, because what we found was they tended to avoid the high silicon plants, the black bars again, because it affected the rate at which they could process food. And if you're a sheep, although I tend to think like this as well, the faster you can process the food, the more you can shovel in at once, the better it is. And so they've got to keep, like all ruminants, they've got to keep the food going into their gut pretty fast. So anything that kind of slows down their ability to take bites is not a good thing. And silicon does that, so they prefer to avoid it. So it has so silicon's having lots of effects. It also reduces the herbivore's performance, their growth rate. So this is the growth rate of our armyworm friends again on 3 different grasses. And again, you can see the growth rates are a lot higher on plants with low silica. And the reason for that was that on low silicon plants, the herbivores could absorb more of the nitrogen in their food. So plants aren't very good food for animals, actually, because animals are mostly nitrogen with a bit of carbon, because we're mostly protein. Mostly fat in my case, but anyway, mostly protein. And then plants are rather different. They are mostly carbon, with not very much nitrogen. So the insects are facing a challenge to get enough nitrogen out of the plant. And that means that when they're feeding on these plants with lots of silica, they can't do that. So that has a very bad effect on their growth rate. And why are they so bad at getting nitrogen out of the plant, even worse than usual? Well, the reason is that silicon is very, very hard. So those knobbly little razor blades are really tough. If you think about the time you went down the beach as a kid or something and you had sort of, I don't know, crab paste sandwiches that your mum made you and you got sand in them, it's like that. It's like chewing that, and it has a really bad effect on herbivores' jaws. So what we did was rear some of these caterpillars on high silica plants and low silica plants, and then chop their heads off, because nobody minds if you do that with insects. We didn't do that with the sheep or the voles, but with the insects, we chopped their heads off, and then we looked at the jaws. So this is an insect that's been reared on a low silica plant, and you can see it's got these kind of big jaws and big sort of teeth mark things. So they've got quite big heads, most insects, not because they've got brains, but because they need to have big jaws and big muscles to cut through the grass. So that's got all its teeth intact, but this is the jaw of a mandible of an insect that's been fed on high silicon, and you can see it's got not very much, needs dentures basically, it's got nothing left here, and this happens quite quickly. This happens in about 48 hours. So when they're reared on high silica plants, you can see the mandible wear is much greater than on low silica plants. So silicon, I hope I've convinced you by now, just surrender, it's easier. Silicon's a really effective defence. It deters feeding, reduces nitrogen absorption, and reduces the growth. And the mechanism for that is probably abrasion. All those sharp, knobbly bits wear down herbivores' mandibles and teeth and make the processing of food a lot more difficult. And we can measure abrasion by looking at the sort of depths of the grooves that the phytoliths and nobbles can kind of cut into plastic, and we can see that high silica plants cut much deeper grooves. So we know it's abrasion. But we don't know the whole thing, because what's really quite odd is that plants can have similar amounts of silicon but be very different in their abrasion, even if they're quite closely related plants. So here are 2 plants that look very similar, because they are very similar, Festuca ovina and a closely related plant called Festuca rubra. And here's their silicon levels. So Festuca rubra is there, Festuca ovina is there, so they're pretty similar. They've both got about 3% silicon, but you can see one of them, Festuca rubra, is not very abrasive at all, and Festuca ovina is much more abrasive with the same amount of silicon. So why do some plants manage to make their silicon much more effective weapon than others? So that was one of the questions we were interested in. And the other question is, why are species so different in their response to attack by herbivores? So this is some work we did when we looked at silicon concentrations after leaves had been damaged repeatedly and after they'd been damaged just once. And some of the white bars are plants that haven't been damaged at all. The grey ones, light grey ones, are ones that have been damaged by scissors. Darker grey is locusts, and really dark grey, black in fact, is voles. So 2 things strike you immediately. One is that a single damage event does not cause an increase in silicon content in the plant. So a plant only responds to attack in terms of producing more silicon or taking up more silicon if it's attacked more than once. And then the other thing that you'll notice is that although mechanical damage does produce a bit of an increase in silicon content, it's nowhere near as much as the response to real herbivores. So the plant can detect when it's being attacked by real herbivores, and it takes up more silicon as a result. But what was interesting is that different plant species do that very differently. So here's one of them, Avanella, and on the right-hand side, that's right, damaged plants, and the left-hand side control plants. And you can see that generally, these are different genotypes of this species. Generally, not much happens. But occasionally, one genotype does respond with induction of more silicon. Whereas June15, which is a very well-defended plant with lots of silicon, generally responds very, very well to silicon addition. But even here, there are some genotypes that respond a lot less, say that one and that one. So there's a lot of variation. So why is that? Well, to know why that is, we've got to know a lot about silicon uptake, which is slightly unfortunate because, amazingly, we still don't know exactly how silicon gets into plants and how it's distributed. The best study we've got is on rice, and that has a fairly unusual root structure, like this. So here's this little root hairs going into the soil, and here's the xylem, which is one of the vessels that goes up the plant. So silicon is taken up from the soil by a transporter called LSI1. And then it moves a little bit through the root tissue with another kind of transporter, there's a kind of pump transporter called LSI2. So far so good. And then it's got to get into the xylem, and there's another transporter to do that, but we don't know what that's called or how it works. Then the silicon goes zooming up into the top of the plant from the roots into the shoots, and it comes out of the xylem and starts to make its way into the leaf cells with a transporter called LSI6. I don't quite know what happened to LSI 3, 4 and 5, they seem to have fallen by the wayside. And then somehow it gets into the leaf cells and is deposited with another transporter that is also currently unidentified. But apart from that, we've got it all sewn up. And then when you look at how the silicon appears on the different plants, you can start to see why some plant surfaces are very, very different to others, and why insects might find these a very different thing to be eating. Okay, so the first thing we can ask then now is, what do plants do with their silicon? Why do plants deposit silicon in different ways? So what we did was to take our two plants that had very similar silicon levels, Festuca ovina and Festuca rubra, but very different abrasions, and our very well defended plant, June15 cespitosa, and we grew them under different treatments. They had no damage or no silicon, they had damage but no silicon, they had extra silicon given to them but no damage, and then they had both. And we analysed the silicon with a new method that we've developed at York, portable X-ray facility, which is really makes it a lot easier. And then we used another kind of X-ray connected to a scanning EM to actually see where the silicon was. So I'll just show you some results now. This is what the plants look like when they've got no extra silicon and they haven't been damaged. So this is June15 cespitosa. You can see it's got lots of really vicious looking spines, and the yellow colour is silicon. So you see they're very silicon rich. This is Festuca rubra, which has silicon all over its surface, but not anything very knobbly or spiny, just a few little round things. Whereas Festuca ovina, this remember is a closely related species, is nothing on the surface, but it looks like it's got measles. It's got hundreds and hundreds of these little knobbles everywhere. And then when you give them extra silicon, they deposit it in very different ways, these different grasses. So June15 says, thank you very much, I'm going to put more on my surface, I'm going to have even more of these vicious looking spines, and just for good measure, I'm going to have some extra little spines as well. And Festuca ovina rubra says, well, actually, I don't think I'll bother doing anything very different to what I'm doing now. I might have a few more of these little knobbles since you asked, but otherwise, no, not really. Whereas Festuca ovina, when it's got extra silicon, covers its surface, still got its little measle-type knobbles, but it's also got some vicious-looking spines, which only appear when it has more silicon or actually when it's damaged, tends to produce more spines as well. So you see, plants have been damaged as well, so you can see the spines are pretty vicious. So these plants have very different ways of deploying their silicon defences. So it's not how much silicon you've got, but what you do with it. So you can see that Festuca ovina and Festuca rubra, they actually have similar silicon levels, but very, very different ways of depositing silicon and very different effects on the mandibles. So this is Festuca ovina, grown on the low silicon medium, so it contains very little silicon, but it causes almost as much silicon wear as another grass species called lolium with much, much more silicon. So it's not how much you've got, it's what you do with it when you've got it. It's a recipe for life, really. So damage and silicon affect silicon content, but plants have different strategies. And Festuca ovina is a much more effective user of its defences. It's much more abrasive with less silicon. But where does that get us in terms of actually protecting our plants and understanding what's going on to improve the way that they deposit silicon? Well, of course, we can actually breed for this trait. And that's a project that I've got going at the moment with the world's largest grass breeders, which you may not have heard of unless you're into grass breeding, but that's DLF trifolium. And they're particularly interested in this grass, tall fescue, so yet another fescue. And it's a turf and forage grass, so it's grown to sometimes feed to cattle as a forage, and it's also grown for things like football pitches and golf courses and so on. And they grow different types. They grow very soft ones for the cattle and very tough ones for hard-wearing turfs, but not too tough or they cut kids' knees to ribbons. And they're very, very interested in understanding how silicon is taken up and deposited in this species and whether they can breed for it. Because they've got different varieties of fescue and they feel very different. So they call these, this is a picture of the surface, you can see it's quite spiny, and they call these different varieties soft, or very soft, or harsh, or very harsh, or occasionally very, very soft, or indeed very, very harsh. So it's not the most quantitative way of assessing how the leaf surface is differing between these different varieties. And when I asked them how they actually actually assigned things to soft or very soft or harsh, they said, we just walk along in the glass house with our fingers, and if we go, ow, it's harsh. Now, that literally is how they do it. So they were quite interested in understanding a little bit more about the leaf surface and to what extent that it was due to silicon, and then whether they could select for either high uptakers or low uptakers. So that's what this project's all about. So here are a couple of varieties, and they've got literally 100 and 100 of these. But this is, the green bars are one of their harsh varieties, and the brown one's one of their soft varieties. And these are our usual treatments of control with silicon, with damage and both. And you can see two things strike you straight away. Firstly, the harsh one is has much higher levels of silicon, and also the harsh one responds to silicon addition and also to damage in the presence of high silicon. So the harsh one has more silicon, takes more up in response to damage. So far so good. What do the leaf surfaces look like? Well, just the same kind of thing as you've seen before. This is the harsh one. Very high density of spines, particularly on the leaf edge, which explains why they go ow when they touch it. And the soft one, there's very hard to see any spines at all. But what we've done is look actually at the efficiency of silicon uptake. So this is what the end result is, the sort of way that the silicon's deposited, but how does the efficiency of silicon uptake alter? Well, here are our categories. Very, very soft, semi-soft, semi-hard. And this is the rate at which they can take up silicon, because it's the total leaf silicon per gram of root material. So it's a measure of how effective they are at slurping the silicon out of the soil. And you can see that there's almost a kind of exponential change here. The harsh ones take up far more. And that's also been known in rice. So getting to crops now, I know forage grasses are exciting if you're DLF trifolium, but they're not that exciting if you're anyone else, in fairness. But rice is a bit more exciting, and here are two varieties, Nipponbar and Kalasaf. And this is exactly the same trait. They've measured the same trait in these two varieties. This is the rate of silicon uptake per root biomass. And the Nippon bar takes up a lot more. So it gets to a higher plateau level than the other one, and it takes it up much quicker. This is a faster sort of kinetics. So then we think, well, is this because this variety has different kind of transporters or in different location? So these are sections of the roots, and the red dots are the transporters. So no, it's the same transporter. It's roughly in the same kind of places. It's slightly more common. There's slightly more transporters in Nippon bar, but the real difference is the efficiency of the transporters, because the gene expression associated with the transporter is a lot greater. So the genes for the transporters are turned into transporters from the DNA a lot more in the Nippon bar. So it's got a more efficient transport system, and it takes up more silicon. And we know a lot now about the genetics of that process in rice. We know the main transporter is LSI1, and genes, the rate of gene expression of the LSI1 gene controls uptake, and that genes with a mutation in this key gene do not take up silicon. So this is the wild type, and this is the silicon content of the mutant, and it can't take up silicon. It gets very, very badly damaged by pests, and its yield is really low. But we know that if we remove the LSI1, then we see very bad effects. If we add LSI1 genes to cells, they take up more silicon. So we know we can engineer crops to be more pest and disease resistant by increasing the rate and efficiency at which they take up silicon. And this has been done by myself and some colleagues in Canada. And this is soybean. Our soybean does contain silicon and silicon transporters, but it's not the most efficient species. It's an important crop, particularly for animal protein, and it's threatened by a lot of diseases, particularly stem and root rot, phytophthora. But when it's grown in the presence of silicon, it does much, much better. So here are plants without silicon, exposed to the stem rot, and then they will just die. And this is the same plants exposed to the same disease, but grown in silicon, and they look really nice and healthy. And what we can do is create plants that have extra transporters put in from wheat, which is a good silicon uptaker, gratifyingly for us, because that's one of our big pea crops, and horsetail, which is not something that we eat, a very ancient plant, but it does contain a very efficient silicon uptaker. And this is the amount of disease in plants grown with silicon and without. And you can see the transgenic plants, they have very low incidence of disease when they're given extra silicon, because this efficient transporter takes it up and defends the plant. This is a susceptible variety without any genetic engineering, and it has higher levels of disease, although silicon still helps. But this is the interesting one. This is a variety of soybean that has no extra tinkering with its silicon, but is resistant to the disease in any case. So two things to note there. One is we can improve the resistance to disease with genetic engineering, but do we need to bother, given that we already have a natural variety that is really resistant anyway? These are just the pictures for you. So this is the susceptible line, still does a bit better with silicon. These are the transgenics. But look at the resistant line. Does really well no matter what. So I love this. This is this should be the title in all my papers. Silicon leads the way. So there's a lot of promise here, and we don't necessarily need to use GM. But why are we so interested in genetic diversity and gene manipulation? Well, to return to our problem that we mentioned earlier, that we really only eat about 10 to a dozen plants. And we have cultivators since the beginning of agriculture, about 7,000 species, and we've reduced that to 10. So we've got rid of an awful lot of genetic diversity. So this is kind of like all the genes in wild species, then we domesticate and lose lots of genes, so it's a kind of bottleneck, and then when we take our early domesticates and make it into a modern cultivar, we lose even more. So this is barley, wild ancestor of barley. This is an early domesticate from about the 8th century. It's only found now in Orkney, which probably tells you something about Orkney or something. And this is a modern cultivar of barley. And more of that in a moment. But the point of trying to do this is if we can reverse some of this genetic loss, we might be able to increase resilience, not just to pests, but also to things like drought. So we can, as I say, do that with kind of a GM approach, and indeed they're increasingly popular. There's more and more GM crops in the world, and interestingly, more and more, we've passed a sort of tipping point really, where there's more GM crops being grown in so-called developing countries than in so-called industrial ones. So, but as I say, we don't need to go there. We can use plants' natural ability. And I've looked at the way that barley's defences have changed over the course of domestication. Now, of course, there's only one defence I'm interested in, and that's silicon. And I wanted to know whether modern barley and ancestral barley were protected by silicon to the same degree. So here's our barley leaf. Thank goodness we don't eat the leaves. These look pretty horrendous. That's a scanning EM, and when we connect it with an EDX machine, we can see the blue colour, varied a bit now, it's not yellow, it's blue now, is silicon. And you can see when you zoom in, you can see the silicon in between the spines. The spines are very silicon rich, and if you do a cross section of the leaf, there you can see very high silicon in the spine and in the cells immediately below it. If we zoom in a bit more, here's it, just the EM version, and there you can see particular high amounts of silicon in the tip of the spine, and again in the cell underneath. And here is a cell completely filled with silicon. So barley's defences are alive and well. What happens if we look at, see if it can induce, just like before, that's with no silicon and no damage. When we add more damage, it deposits more silicon. When we give it silicon, we see even more. And then when it's damaged and it's got silicon, it just fills up the whole leaf right across. So it's filled in the gaps between the spines. So the whole leaf now is covered in silicon. And interestingly, when we look at our kind of Orkney variety of barley, our early domesticate, and compare it to our modern cultivar, they are very similar. This is our modern cultivar, this is our early domesticate, and you can see there's a little bit more silicon in the early domesticate, but not much. And both of the cultivars respond to damage and to silicon addition. So our defences are alive and well. So there are some crops that haven't lost their defences through domestication, and that gives us some promise. And I want to spend just the last 5 minutes, and that's all it is, because now we're running out of time, on another project that's using the same sort of techniques to try and make plants more resilient to drought. So I've talked a lot about improving resilience to pests. That's my sort of main area of research, but I'm also working on making rice in particular more resilient. I have worked on barley and drought, but I'm most interested in rice and making it more resilient to unpredictable water supply. And this is a project funded partly by the UK government, Department for International Development, and the Bill and Melinda Gates Foundation, and the Indian government. Because what we're trying to do is we're trying to get back some of that genetic variation that we've lost when we domesticated rice. Remember I told you that this variety, the Rhiza sativa japonica, has 10,000 fewer genes than its wild relative. So here is its wild relative. It looks more like a tree than rice. So Rufi pogon has lots and lots of roots, lots and lots of leaves, not very much grain, so it's not much use in terms of yield, but it is wonderful at resisting the sorts of stresses that kill off our more pathetic modern cultivars. So what we can do is, well, we can, I say we, if we're an ace rice geneticist at Cornell University in the US called Susan McCooch, what we can do is cross this unlikely looking specimen with modern cultivars, and we're using one called Karinga, but we're also going to use one called IR64, which is the most popular rice variety in India. And by doing lots and lots and lots of crossbreeding, we can produce lines, and here they all are down the side here, roof 47 all the way to roof 48 and roof 27 and roof 23, and these little black dots here all the bits of the rufipogon DNA that have been put into, crossbred into Coringa. Not through genetic manipulation, just through crossbreeding, ordinary crossbreeding. And so we can see that we've got now these lines. So Ruf47 has a bit, it's a Coringa plant, but it's got a bit of rufipogon genome in chromosome 1, a little bit more in chromosome 2, a big chunk in chromosome 3, nothing in chromosome 4 or chromosome 5, and a big chunk in chromosome 6. So we've produced a whole load of these lines, almost 200, that have different chunks of the wild ancestors' DNA. And we know that that's going to change their characteristics. So this is Coringa without any host DNA in, and its roots are really short and feeble, but this is one of these lines with much longer roots. So we know that by adding a little bit of rufipogon DNA, we can get beneficial traits like longer roots, which might make them more drought tolerant, but, and this is the key thing, it will still have the yield benefits associated with modern cultivar and varieties. So the idea is to maintain yield under drought using beneficial traits transferred in from the wild relative. And then we test those using our field sites at the Indian Rice Research Institute, and this is one of our colleagues there, Nina Swain, with our first lines in the ground. Very exciting. We need to understand the mechanism so we can inform plant breeders what traits they're looking for. So here at York, we're in Cardiff. At York, we're growing all these different lines in carefully controlled conditions and looking at their physiological traits and metabolites and gene expression. We're doing things like looking for changes in response to drought. So the shaded bars here are plants that have been droughted for 7 days at a key stage. This is Coringa. Doesn't respond to drought by doing anything with its root fresh weight, useless. This is line 22, which has got some rufipogon DNA in, but obviously not the right bit, because that doesn't respond either. But line 4 is looking a lot more promising. In response to drought, it produces a big increase in its root length. That's what we need. That's what we then tell the breeders about and help them generate new varieties to trial. And the key thing for us is to identify the changing traits above Coringa that are beneficial and link those to particular segments of the DNA. So here are a whole load of lines tested for the number of tillers they produce. Here's our Coringa, our elite, so-called elite cultivar. And here are the improvements in the different rufipogon lines. So the best one here is number 27. One thing to note though, some of our lines are worse than the original. So just because we've put a bit of wild ancestor DNA in doesn't necessarily make any improvement. So these ones are going in the bin, and we're focusing on these ones, and we say, right, what has 27 got that no one else has got that might explain this trait? So we look for the right segments in rufipogon 27. So that's how it works. What do we do with them? Well, we need to know where the best new varieties can grow. So in India, 18% of the rice growing area is prone to drought, so this is a really important problem. 45% of the rice harvest in India is rain-fed, so that means that the farmers don't irrigate, they're entirely dependent on the monsoon, which turns out to be a bit of a problem because our historical data, when we look back at climate data and plot the changes in monsoon rainfall against rice yields, we see that each additional drought day, or day within the monsoon period where there's no rain, each additional, so it's not really a drought, it's just a day without rain. Each day without rain reduces their yield by 9 kilos a hectare. And if you're a subsistence farmer in a poor area in India, that is not good news. And I'm back on my depressing mode now because there's even worse news. When we look at our climate models, looking at areas that will become unsuitable for cultivating rice in this way by 2050, about half the current areas will not have a climate that supports rain-fed rice production unless things change. So you can see all these orange areas currently grow rain-fed rice, but the climate is likely to become unsuitable, the monsoon too unreliable. So that's a concern for farmers. So our social scientists are working with farmers to find out about what will help them in terms of taking up some of the new varieties that we produce. What are they looking for? And it's been very challenging, but a big part of the team, this is a big effort at York and at Cornell and in the Indian Rice Research Institute, is to talk to women. Very hard actually to get access. That the men don't like you to talk to the women without them present, etc. But we've worked around that with Indian women PhD students and so on, and we've got some information about what's important to them. They're the ones who have to feed their families. They're the ones that do most of the work on the farms. And the key trait for them, they were very interested in drought resilience, very interested in pests, but they kept saying, Dr. Sue, don't make them any more work to harvest because we have to do all the work. So we're looking at all the kind of aspects of changing cultivation patterns. And that's what we need to do for a sustainable future. We need a second green revolution, which is knowledge intensive rather than input intensive. Not to say we aren't going to need inputs, but we need to use our brains, not just our chemicals at the moment. So how are we going to increase innovation? Just a quick mention for a new programme that HEFCE, that's the Higher Education Funding Council for England as opposed to Wales, they've given £16 million to research partnership of all northern 8 universities. And we're looking at how to improve agri-food resilience to ensure the stability and integrity of agri-food supply chains in the face of global challenges of climate change, declining resources, and rapid socio-political transformations. And after breakfast, we're going to find something else to do. Huge challenge, of course, but we've got a good team to do it. And I just want to flag up, I've talked mostly about food production, but that's only a third of the story. We need to look at food supply chains, the integrity and safety of them, and we need to look at consumer behaviour and nutrition. A really frightening statistic is there are now more people in the world who are overweight and obese than are hungry. So we have enough food, the wrong people are eating it. And I include myself in that before. The next stage, so we need to get some innovative thinking, some big collaborative projects, some projects that look at the whole food chain, not just production, but also waste, consumption and so on. And we need to train the next generation. So a couple of training networks at York on biotic constraints to crop production. Obviously that's what I'm involved in, plant interactions with pests and parasites. Our next big doctoral training programme is going to be on enhancing resilience to water stress, so back on the drought. And the other thing we've got to do is work with practitioners in the industry. So the government's funded some innovation partnerships between universities and real farmers, and what are they doing, and working with them to work out ways to improve their natural crop protection. So this is a project on using biofumigants, natural plants that you plough into the soil to improve resistance to pests. So we're working with a big set of 9 different farmers are trialling different products. So practical approaches. And that's the message I want to end on, really. It's innovation, interdisciplinarity, and practical approaches to change. And I think you can't do better, really, than end with a quote from Darwin. The species which survives is not the strongest, nor even the most intelligent. It's the one that adapts best to change. And we've been changing our agriculture ever since we first started doing it. And I believe we can continue to change our agriculture and really make a difference. Thank you.
Thank you ever so much. Do you want to?
No, I was just going to say, I always forget to thank everybody. You know, I stand here and take the credit. Of course, huge team of people involved, lots of different researchers and lots of different money. And the prize for the worst Christmas jersey definitely goes to Pete here. Okay, thank you.